So welcome everybody to an exciting edition of our webinar on emerging topics in biomolecular magnetic resonance. Please don't record. Both speakers of today have agreed to recording, and Kevin Gardner will upload the recorded talks to the ICMRBS YouTube channel. I also remind you that to type your questions into the Q&A box and not into the chat, or raise your hand, and then we can unmute you and you can directly ask your question. There's also the Early Career Researcher Magnetic Resonance webinar, very successfully ongoing, so please tune in there too. Without further ado, I hand over then to the first chair, Hartmut Oschkiner from Berlin.
Yeah, hello everyone. It's an absolute pleasure to introduce Marius Clore. And as you can imagine, somebody more important has been asked to do it. Robert Huber, Christian approached Robert to have an appropriate introduction. And as you can imagine, with such a young, flourishing scientist, he has obligations with his company Proteros and can't do it today. So he sends his best regards via email, and maybe he links into the lecture later. But that brings me also to a good beginning of the introduction. I know Marius since the days in Martinsried, when he had a Max Planck Young Research Group on the same corridor as Robert Huber's office, and that was a very nice situation. All doors were open, and it was very common to discuss things in the rooms with open doors, and especially also on the corridor. And I called it the corridor of scientific rigor, because many discussions were around how to understand things best, how to do them best, and how to progress best, and I enjoyed that very much. Sometimes these discussions got loud, but in all groups, also in the Huber group, and still these were very good times. I enjoyed that very much, I have to say. So that was a good time. These were sort of the heydays of structure determination, structural biology. Also, X-ray crystallography was on a very strong rise, also because of the invention of phase plates and all these things. And in those days, Marius has made incredibly nice contributions. He was one of the first to determine structures, and especially to determine protocols and software pieces to calculate these structures. And one has to say, nowadays, what we use, I hope I'm not wrong, you may send me bad messages, the software offsprings from the Wüthrich lab, and in principle from Marius's lab, if I may call the CNS editions on the NMR side like this, are the still used ones. So I think that was an incredible contribution. And they're very important structures and other technical developments. I should have said that before he was at the NIMRC in Mill Hill, where he started with the transfer NOE experiment that was also very much connected to his name. What is most important is Marius stayed always at the top or close to the top of the wave and is still riding it. So this is amazing to see, and changed with time. And that is also quite interesting with regards to how the field has changed. In those Martinsried days and later, very important structures were made. I think about interleukin beta 1, interleukin 4, and other things. And the idea at that time was to do the structures as quickly and as good as possible. And later, when I visited the NIH, Marius said live in front of the camera, my camera, We don't do structures, we do biophysics, and he does that incredibly well. I liked very much the work using paramagnetic probes to describe the dynamics in biological processes like proteins writing on DNA and things like that. So he stayed very much on top all the time, and I think this is an amazing career which would deserve more as a prize than this lecture here. And with this, I would like to hand over and give the microphone to Marius.
Okay, well, thanks, Hartmut, for that really nice introduction. Takes me back over 35 years, I guess, since I left Germany. So interesting times then. And let me also thank Marcus and the others for inviting me to speak. So let me just start, let me share the screen. So what I'm going to be talking about today is actually using NMR to probe transient sparsely populated states. And I'm going to focus on a protein or a portion of a protein called Huntington, which is responsible for a rather nasty disease known as Huntington's disease. It's a fatal neurodegenerative condition. Many of you are probably too young to know that there are some rather famous people who actually developed Huntington's disease. And one was the guy who inspired Bob Dylan. Maybe Bob Dylan is, you guys are too young for Bob Dylan, but a guy called Woody Guthrie inspired Bob Dylan, and he actually died early from it. Let me also acknowledge the various people who've been involved: Alberto Chicon, Sam Kotler, Mario Walti, Thomas Schmidt, Charles Schwitters, and Vitaly Tugurinov. So what we've been focusing on in the last, I don't know, 15 years or so is techniques to probe states that are very sparsely populated and transient, that are effectively invisible and basically impossible to study by conventional biophysical techniques. So that includes crystallography, conventional NMR, single molecule spectroscopy, and even cryo-EM. And everything relies basically on something that really goes back to the transferred NOE in a sense. It's to transfer some sort of relaxation property from an invisible state to an NMR visible one through some sort of exchange process where one can observe the effects on the easily observable NMR visible species. And the types of things one can use are paramagnetic relaxation enhancement. It's based on a 1/r to the 6th dependence, so it's extremely sensitive. The effect is very large. And you can detect minor populations, let's say at a population of about 1%, maybe a bit less, in systems that exchange with lifetimes less than, let's say, about a half, 250 to 500 microseconds. So that's based on distance. Then there's relaxation dispersion spectroscopy. So Lewis K, R. Palmer, I misspelled Peter Wright there, Peter Wright and Jane Dyson have played a very important role in developing some of this. And this is based on differences in chemical shift causing chemical exchange line broadening and probing this and getting kinetic information out of that. And then the last set of things are dark state, and there are variations on this, a dark state exchange saturation transfer and lifetime broadening, which rely on very large differences in molecular weight between the NMR visible species and the NMR invisible one. And the NMR visible one can basically be in the solid state regime, providing it's suspended in the NMR tube. So now I'm going to focus on Huntington, and the key element here is the portion that's encoded by exon 1, which comprises 3 regions, a short N-terminal amphiphilic sequence, a polyglutamine sequence, and a protein-rich domain. When the polyglutamine sequence exceeds 35 residues, you're in bad shape. You're going to die pretty young from a really nasty disease. So our idea initially was to look at a construct comprising the N-terminal domain and the minimal length of Qs where the peptide would still form fibers after a while, but would hang around long enough in a monomeric state to be able to do some NMR. And the thing that we settled on initially was this one, which I'm calling Q7, which has 7 glutamine repeats after the N-terminal region. Now, if you have longer repeats, it really aggregates quite fast. I mean, this is at 5 degrees. So even after, after 100 hours, there's nothing left if you have Q14. But this Q7, after, let's say, 5-6 weeks, will form fibrils. And this is an AFM picture. So let's see what we see just qualitatively from the NMR before going into anything quantitative. So let's just look at REX terms from C alpha CPMG. If we just take the N-terminal NT domain at high concentration, it's basically flat. But as you raise the concentration of the Q7, you get large REX terms. You also get large exchanges in exchange-induced shifts for the C alpha. Nothing really happens to the NT domain on its own. Then what about increases in lifetime line broadening? Well, not lifetime line broadening in this case. Increases in R2 or R1 row. If we just take the NT domain at dilute, which is gray, or concentrated 1 millimolar, there's no difference. If we take a dilute sample of Q7, so at 10 micromolar, that's green, or a methionine oxidized, so a sulfoxide form of this peptide at 1 millimolar, there's nothing, but you take 1 millimolar of the native species, you see all these large effects. So something is going on. Well, what is it and how can we analyze it? And to cut a long story short, what we basically did is we analyzed a large data set globally, fitting various global kinetic parameters and obviously residue-specific parameters such as chemical shifts and R2s. And what we had is we had C13, we had C alpha and N15 CPMG relaxation dispersions at a number of concentrations, exchange-induced shifts over a whole range of concentrations from 50 micromolar to just over 1 millimolar. R2 dependence, concentration dependent R2s from 50 micromolar to 1.2 millimolar, and then some R1 row dispersion. And these are just some examples. So this is relaxation dispersion, exchange induced shifts, N15 relaxation dispersion, N15 R1 row, N15 exchange induced shift concentration dependence, and R1 row concentration dependence. So we can take all of that data, and this is just a subset, and fit it all simultaneously in the minimal model that can be used to fit the data is actually a 4-state scheme. So we have monomer, which is approximately 95% of the, well, these little percentages here is what's happening at 1 millimolar. But the predominant species, the NMR visible species, is the monomer, and you have a branched oligomerization pathway. So you have a slow off pathway leading a slow off-branch pathway to a dimer. I'll go on to what it is that basically doesn't go any further. And then a productive pathway, which is on a faster time scale, which leads to a coil-coil dimer and eventually a coil-coil tetramer. And we can get all these rate constants and equilibrium constants and so on. Now, what is the structure of these productive dimers and tetramers? So first, just qualitatively, we can just look at the concentration dependence of the C alpha and C beta chemical shifts. And what we can see is that all the C alpha shifts become positive, the C beta shifts go negative. So you know straight away that at least for residues 3 to 17, so that's from here down to here, you've basically got helix being formed. After that, there's very little helical content. And before that, there's basically no helical content. So from all the fitting, we get what the C alpha delta omegas are for the on pathway, and that's in red, for the off pathway in blue. So the off pathway is partially helical. So let's say it has increased helical propensity and presumably comprises an ensemble of dimers that are in partial register or different sorts of registers. And then you have something that's fully helical from about that's the on pathway species, which are both dimer and tetramer. In this case, we assume that their shifts are the same because these are secondary shifts, so they'll be dominated by secondary structure, not by quaternary structure, are essentially entirely helical. So it's entirely helical from residues, let's say 3 to 17. So what about the structure? We know that 3 to 17, so, and the population of these species is, of course, very low. It's about 2% or so. Well, we can look at intermolecular PRE data. So we have 2 paramagnetically labeled samples that are not labeled with N15 or C13. One at C12, we put a nitroxide on it, the other at S15. And we look at the effects on N15 labeled on the N15 labeled peptide, and we see these up and down patterns. So there's clearly structure going on. This is indicative of helix. It's indicative essentially of both anti-parallel and parallel conformations. And we can do controls to make sure, which are shown over here, that these things have got nothing to do with interaction of the nitroxide with the N15 labeled peptide. These are due to peptide-peptide interactions. So given that we know where the helix is, we can then use rigid body simulated annealing to refine directly against the PRE data to get structures. Now it's a little bit complicated because we've got a paramagnetic label, and the paramagnetic species is actually only about 10. We only, the ratio of paramagnetic to diamagnetic is 1 to 10 to 1 to 15. The problem is that once you attach a paramagnetic label, you change the dimerization and tetramerization properties. You actually enhance things a little bit, so you get more dimer and tetramer. So we don't know exactly how much dimer and tetramer it is, so we have to do a grid search where we have varying proportions of dimer and tetramer. And the bottom line is that the structures, you get the same structures, providing you have at least, let's say, 30%, more than 30% tetramer, and less than 70% dimer. After that, the PRE-Q factor goes up, the PRE correlation coefficient goes down, and the agreement between observed and calculated PREs is extremely good. So what you basically have here is an anti-parallel dimer shown in red and in blue, so two helical coil-coil dimers that form a dimer of timers, and this is the tetramer. So what does the structure look like? Well, the dimer has a very nice hydrophobic interface comprising leucines and phenylalanines, and it's C2 symmetric. That's actually constrained into the calculations. The tetramer is D2 symmetric, and again, we have a hydrophobic interface, but I just draw your attention to this methionine 7, because if you mess this up and make a sulfoxide, you basically block tetramerization, and hence you don't get fibrillization once the methionine is oxidized. And it's interesting to note that this structure happens to look very much like the P53 tetramerization domain, a structure that we solved now almost 30 years ago. We can actually validate the structure using, well, I can't partially see my screen because it says screen is shared, but this is a construct comprising a GB1 domain fused at the N-terminus of the Huntington with S15C. And this particular construct actually only forms a dimer, it doesn't go on to the tetramer. And you can see that by, we can do inversion modulation DIA, and if you do this, you can fit it and it only goes to the dimer. You can get the KD of the dimer, which is considerably tighter than the dimer in the absence of labeling, and you get a distance distribution out of the DIA data, and you end up with a distance between S15, between the nitroxide labels in this particular construct within the dimer of about 26.5 angstroms or so, or about 27 angstroms, well, 27 to just below 28 angstroms, and that agrees very nicely with what you get experimentally on that structure, which is about 26.5 angstroms. So what does this tetramer mean, and why is it important? Well, what the tetramer basically does is it brings the polyglutamines of different subunits into close proximity. So what it's doing is it's effectively increasing the local concentration, if you will. By increasing the local concentration, you increase the propensity to nucleate and form polyglutamine fibrils. And the closest distance of approach is between dimers of the tetramer. So for example, between this red here, the C-terminus of this red subunit, and of the blue of this blue subunit. So the anti-parallel within the dimer, the distance is quite long, and the anti-parallel distance within the tetramer is also quite long. So the shortest distance is the parallel one within the tetramer. And if there are enough glutamines, then you get aggregation very rapidly. So the next question is, how can one inhibit nucleation and fiber formation? And there are obviously a number of ways you could do it. One, you can just mutate within the N-terminal region to prevent tetramerization, or you can modify things within the N-terminal region, and I already alluded to it, because you can oxidize the methionine, and I'll show you in a second that you can actually do that with titanium oxide nanoparticles. Or you can just sequester the monomer by chaperones such as HSP60. You reduce the concentration of free monomer in solution, and therefore you increase the critical concentration, the effective critical concentration that's required for polymerization. Or you can have an indirect allosteric mechanism, which I'll talk to at the end, which is actually binding to the polyproline rich domain and messing up oligomerization that way. So very quickly, there's a paper published in JACS. It's sort of interest. If you take titanium oxide nanoparticles and activate them with light and add them to Huntington, that will oxidize the methionine and it will reduce the amount of aggregation. So if you look at the aggregation just by looking at the decrease in intensity of the NMR visible, let's say the NMR, the amide envelope, if you just have, say, the Q10 species on its own, well, it eventually disappears completely. Whereas with the titanium nanoparticles, it plateaus at about, in this case, 35%. And that simply has to do with the amount of hydrogen peroxide that's generated here. More hydrogen peroxide, more titanium nanoparticles, you can go all the way and prevent it completely. Now what's interesting about the titanium oxide nanoparticles is that you can follow the oxidation of the methionine, and these particles accelerate or catalyze the oxidation about 10-fold, 50-fold. So the K1 is 11 molar to -1 hours to the -1 slow units with hydrogen peroxide compared to 500 in the presence of the titanium oxide nanoparticles. But from the NMR perspective, what's interesting is that we can then analyze the interaction of the Huntington peptides with these titanium oxide nanoparticles, both the reduced state, the oxidized state. We can do DEST, where there's clearly a dual component in there, so there are obviously two species. Then you have lifetime line broadening because it's binding to something large. And to cut a long story short, what you have is you have a species that's bound, if you will, that's partially restricted, so it's within the envelope of the nanoparticle, but not directly bound to it, so it has a bound R2 of about 500, and then a fully bound species where the R2 is basically 31,000, so this is effectively like a solid state particle. You have hydrogen peroxide oxidation, which is accelerated in the presence of the titanium nanoparticles, and it's nice that NMR can characterize all these various species and states which proceed, let's say, on the millisecond timescale. Well, let's go to something more biological, and that's the interaction of Huntington with the chaperoning GRO-EL. So GRO-EL admittedly is an E. coli protein, but mitochondrial HSP60, human mitochondrial HSP60 is over 55% sequence identical. So it's basically the same. GRO-EL is a macromolecular machine. It's a 14-mer, it's about 800 kilodaltons. It comprises 2 7-mer rings, which have a very large cavity that can bind substrates. So if you take fluorescently labeled Huntington peptide with 14 repeats, and you do this in the absence of GRO-EL, you get aggregation. So that's what, this is a THT assay. So this is what, fluorescence assay. So this is what's shown here. If you add GRO-EL, basically nothing really much happens. Now you can also do this with a mini chaperone that just comprises it's monomeric and just comprises the apical domain of one of the subunits of GRO-EL, and now we've got a THT assay. When you have no mini chaperone, so the blue, you get aggregation. When you don't have mini chaperone, when you have mini chaperone present, nothing basically happens. So if you look at this with what we have with GRO-EL, in the absence of GRO-EL, you get fibers. In the presence of GRO-EL, you get some dirt, you get no fibers, but all these little things that you see here that are far too small to see on the screen are actually GRO-EL particles, which are pretty huge. In the case of Q10, in the absence of mini chaperone, you get fibrils both by EM and AFM. I'm not showing the pictures in the absence because the screens are blank, so the grids are blank, so except for dirt, so there's nothing to show there. Well, where do they bind? We can do some trivial things. We can do chemical shift perturbation. Where do they bind on the mini chaperone? Well, we can do chemical shift perturbation on the mini chaperone side. We can look at intermolecular PREs as well. So you have paramagnetically labeled Huntington peptide and unlabeled and 15 labeled GRO-EL, and you see predominantly regions 1, 2, and 3 that correspond to these two helices here and this little helix here, and these are known to be binding sites for substrates. And if you do the reverse experiment just with chemical shift perturbation, so something really trivial, all the perturbations occur predominantly in the N-terminal region and then tail off with the glutamines and with more glutamines, for example, with this Q14 and then P11 at the end. Basically, nothing happens in this region here. So it basically ends at residue, let's say 17. So the region that's binding is predominantly in the center, is predominantly at the N-terminus. We can characterize these by relaxation dispersion in the case of mini chaperone and exchange-induced shifts. For intact GRO-EL, then we have to use lifetime broadening and DEST as well as some relaxation dispersion. If we just concentrate on this, so this is Q7, both binding to the mini chaperone and to the intact GRO-EL. The KDs are about 1.5 to 1.7 millimolar, so 1 to 2 millimolar. The rates are quite fast. The dissociation rate constant from GRO-EL and the association rate constant are down by a factor of 2 relative to the mini chaperone. That's easily explained because you probably have to have several tries to get to hit the substrate binding site within the cavity. And then if it dissociates within the cavity, you can get geminate recombination. So that would slow the overall thing. If you look at Q14, P11, the KD is more or less the same. The rate constants are more or less pretty similar with the Q7. The analysis is a little bit more complicated because Q14, P11 also undergoes tetramerization. So you have to take that into account, but I won't go into details with how that's done. You just create a virtual species to account for that. And then the final thing is how can we inhibit fibrillization and toxic effects allosterically? So profilin, which is a very abundant intracellular protein that binds to polyprotein repeats, has been shown in these two papers up here. Rohit Papu is the latest paper. It inhibits fiber formation and reduces toxicity of Huntington exon 1. And so what's going on? And what is the mechanism? So we looked at the first we looked at a longer construct now that comprises the full exon 1, but again with a Q7 repeat. The mechanism exactly the same, off pathway leading to a non-productive dimer, on pathway leading to a productive tetramer. The rates are a little bit smaller. There's one little NMR trick that I would just simply draw your attention to. So this is all based on the same experiments that I described properly, but we weren't able to determine, we were only able to determine a lower limit for K-2. Now you can resolve this by simply looking at the concentration dependence of R1 row that's shown here and multiple spin lock fields. And when you do that, you can actually determine exactly what K-2 and K2 is. So that's just a neat little NMR trick. What about the interaction of profilin? Well, in terms of mapping, profilin, the region of Huntington that binds to profilin mapped by chemical shift dispersion is in the proline binding region, and the two regions that bind to the Huntington are the two polyproline tracts whose resonances, the C alphas, are completely broadened out, and then you also see shifts N-terminal and C-terminal to both polyproline tracts. So the two regions are the polyproline tracts. You can analyze the concentration dependence, get the KDs. The KD, the binding to the first polyproline is about 2-fold tighter than to the second, and getting 2 molecules of the Huntington on to the, the profilin on is negatively cooperated by about an order of magnitude. But the key here is the following. If we now look at the concentration dependence of the exchange-induced shifts, if we take just the free, right, we see concentration-dependent exchange-induced shifts for the Huntington peptide, both N15 and C-alpha, so due to helix formation at the N10 of the N-terminal region. If you add profilin, that's completely abolished. However, when you look at CPMG relaxation dispersion at concentrations where you wouldn't see relaxation dispersion at concentrations where you wouldn't see relaxation dispersion, basically you now observe relaxation dispersion. So what's actually going on? Well, what is going on is that the productive pathway, so this is what's going on in the free. So this is the gray, non-productive, productive. The productive pathway is completely blocked. No productive pathway occurs. The productive pathway contributes mainly to the R1 rho and the C alpha, and to the later phases of the CPMG. But the CPMG effects that you observe are due to the much slower effect, or the initial phase of the CPMG is due to non-productive dimerization. So in effect, what you have, and this occurs on a time scale of about 600 microseconds. So what in effect you have is you have something binding to the polyproline rich region, which is quite a way away from the N-terminal region that oligomerizes. There's a polyglutamine stretch in between, and it affects the oligomerization properties of the N-terminal region such that you no longer form a productive tetramer, and hence you effectively, you block the productive pathway completely, and consequently you no longer get nucleation and fibril formation. So basically, in conclusion, what I've shown is that we can monitor, so we can look and dissect sub-millisecond events leading to pre-nucleation oligomerization of Huntington. While, biologists may think these are irrelevant, they're actually extremely important, because tetramerization is absolutely the key to subsequent nucleation and fibril formation. And if you inhibit that tetramerization that occurs on the sub-millisecond time scale, you basically knock out fibril formation. And, if one could do that in vivo, of course, then you could cure the disease, or at least prevent the onset or delay the onset of the disease, which is, of course, pie in the sky, because that's very far away. But be that as it may, it provides a theoretical avenue for dealing with this. And with that, I'll be happy to take any questions.
Thanks a lot. You're ready, Marius. That's what you wanted to indicate, right? Yeah?
Yeah. I'm finished. Yeah.
Thanks a lot for this wonderful talk. May I make a remark to your beginning? Woody Guthrie, for the younger ones, Woody Guthrie composed "This land is your land, this land is my land," so something very important.
That's right. You're showing your age, like me. But we were both too young for Woody Guthrie, though.
Well, they still sing it.
I think he died before either of us were born. But be that as it may.
I'm not going to Wikipedia now. I start in the order they appeared, also because I like sometimes questions to nitty-gritty details. How did you calculate the error bar for the shifts in figure B, carbon 13C, alpha, delta, omega, ppm? I think you remember which slide. I don't.
Well, I'll put it up. I think. I know what you mean. Okay. Well, I mean, that's from the. You mean from this stuff here, right? You mean from this?
I don't know, the person.
The delta omegas.
Yeah.
Yeah, I mean, that's just from the global fit, because the delta omegas are an optimized parameter.
Mm-hmm. Good. So Walt Maszewski asks, any ideas about the structural difference between the productive and non-productive dimers? Yes.
Well, I think I mentioned it, but I probably didn't make it clear enough. So the productive dimer and tetramer, right, are fully helical from the C alpha shifts that you get out from the complete analysis, right, are basically indicative of a fully formed helix. So that's the red stuff here. The shifts for the non-productive dimer, C alpha shifts, are nowhere near as large. I mean, they're like a factor of, let's say, 1.5 one and a half to twofold less. So in other words, instead of being fully helical, it's partially helical. And I suspect what's going on is that you can form the dimer, for the non-productive dimer, you can form it in different registers and also presumably in different orientations. So you could have parallel, anti-parallel coil coils or partial coil coils. You can have partial helices that interact with each other. So they're not fully formed, whereas the real dimer and the real tetramer, I mean, they're classical coil dimers and coil tetramers.
Okay. Victor Lopez asks, most of these interesting experiments were developed using healthy exon 1 from Huntington. Healthy in quotes. Are there any studies of this kind, but using polyQ expanded more than 36? Huntington?
Well, we're not at 36. At the moment, we're doing work. We're looking at a construct, a full exon 1 construct with 35. I can't tell you the results yet, not because we don't have them, because it's a tricky business. Because the problem is that the solubility limit of that construct is extremely low, so you can't really go. You're limited in concentration, maybe to 200 top concentration to maybe 100 micromolar as opposed to 1 millimolar. It's not that you can't do concentration, you can't do experiments at 100 micromolar, that's no problem. But doing experiments at 10 micromolar to look at a full concentration dependence and so on is a little bit touchy. So, that's why we focused on the Q7, simply because it forms fibers eventually, and it maintains solubility long enough that one can actually do these experiments.
Yeah. Zach Boswell asks, hi Marius, great talk. Given this chaperone stabilize the monomeric form of HTT, why do you think they are unable to do so in vivo in the disease? I guess because of how many you have, is it? Or, well,
I mean, that's a difficult question to answer. I mean, who knows what, I mean, the thing is this, right? All these so this is like off the top of my head. So all these chaperones and this whole chaperone machinery obviously does do something, because if it didn't do anything, you wouldn't live to 40, 45, 50, 55, because the pathological constructs in a test tube aggregate within minutes, right? Well, these people are living for years, right? So there is a machinery that's taking care of these. And for example, HSP40 is known to disaggregate fibers of Huntington. But I mean, after a while, right, the system gets overloaded and you're out of luck. This is just guesswork.
There is one or two questions from Anna Chakraborty, and I'm trying to, well, I read it. How do you know that the oligomerization ends with glutamine? Can you keep track of the modification of dimerization made by paramagnetic centers and then calibrate your system? The first one I don't quite understand. Maybe
well, I think okay. I know what well, if I don't answer it correctly, they can correct me. So the thing is that in this particular construct, right? As you increase the length of the glutamines, first there are no shifts, so they're not helical, and second they're mobile, judged by R2 and so on. So and as you progress outwards, you don't get any increase in R2 as you increase the concentration. So basically, they're moving around and free to float around. So you know that they're not part of the productive tetrameric or dimer, the productive dimer and tetrameric core structures. I mean, they're not part of the coil. What was the second question again?
Can you keep track of the modification?
Yeah. Right. So here's the thing. First, we know what we did do, and I didn't show a slide of this, is that we characterized the oligomerization properties of the nitroxide labeled, of various nitroxide labeled constructs. Actually, I might even have a slide of that so I can actually show you later on. Hold on a second. Okay, right here. So what we did is we actually had various constructs comprising GB1 with these nitroxide labels on. And these nitroxide labels, some of them, for example, this GB1, the Q7 with S15C only goes to the dimer stage, both by AUC, by analytical ultracentrifugation and EPR. The one at S12C does form both a dimer and a tetramer. Putting these modifications, both these modifications in, actually increases the affinity of the dimer. I'm not quite sure why that is, but I suspect that anything you do, you can blow on this and you'll change the oligomerization properties, just as you can blow on it by adding profilin and you change the oligomerization properties. But interestingly, the dissociation constant for dimer to tetramer is not really affected by the labels or by these constructs. I mean, it's all, let's say, 10 to 20 micromolar, whereas the others are from, 1 millimolar, let's say half millimolar for these modifications, all the way to 120 millimolar for the wild type. Okay. Now, in terms of what their structures are, I mean, the pattern of PREs is completely consistent with what you would expect for a coil structure. So, I mean, this is not, that would indicate that you are forming that.
Thanks a lot. I think you answered the first question. There's still Gaetano and Walter Chasin, but Mei Hong has sent me a yellow card. We should go on, actually. Thanks a lot for your talk, for a wonderful presentation, and for the very nice answers.
So we move on to the second speaker then, which will be Guido Pintacuda from University of Lyon. I will not introduce Guido because he did a postdoc with Gottfried Otting in Australia. Now for Gottfried, unfortunately, it's a little bit late now. So he agreed to introduce Guido, but recorded the introduction. So I will now play the words by Gottfried.
Otting, and I was Guido's supervisor when he joined my group as a postdoc between 2002 and 2005. In France, the Écoles Normales are well known for educating some of the best and brightest minds who go on to play important roles in public administration and politics. It is less well known that Italy also has such a school, which is the Scuola Normale Superiore in Pisa, which has produced, for example, a fair number of Italian prime ministers in the past. Guido came to me from this school, where he had done his PhD with Lorenzo Di Bari. He was, and still is, an expert in paramagnetic NMR, and I was very fortunate to have him in my lab. Indeed, I learned a lot from him. Innovative and alert, it is little surprising that he went on to build one of the most vibrant groups in solid-state NMR in Lyon, starting as a postdoc with Lyndon Emsley and becoming the head of the solid-state NMR group for biomolecular analysis just there in 2009. Besides having won important prizes such as the Sackler Award and the Star of Europe Award, an important characteristic of Guido's is his almost unending willingness to contribute to the NMR community, such as coordinator of a European network grant. I have no doubt you will enjoy his talk today.
So this introduction, so Guido, it's yours.
Thanks, I'm very touched. Thanks, Marcus. Thanks, everybody. Thanks, Gottfried, for the introduction. So I miss you all. For share screen. Let's see whether this works. And I play that. Can you see my screen? Can you hear me?
Yes,
it's fine. Okay, so let's. It's time to make something to give a good talk after the wonderful talk we just listen to. But so I'll try to bring you some news about membrane proteins and solid-state NMR with fast mass. So Lauren, Andreas has already covered the topic a few seminars ago, and so will be a lot of things you may have already heard, but it's important to hammer some concepts again. So Lauren started this work in when he was a postdoc in Lyon, and now he's carrying it out in Göttingen. So I'll talk about membrane proteins and proton detection and fast magic angle spinning. So you are all aware of the fact that proton detection is the best way to optimize sensitivity in NMR because of a large Euromagnetic ratio. However, in solid state, because of the large Euromagnetic ratio, the strong homonuclear dipolar coupling happening in solids, the proton detection in solids at slow magic angle spinning is possible only with heavy dilution of the proton content, so in a heavily deuterated context with sparse reprotonation. The field has evolved with the availability of faster and faster probes capable of handling smaller and smaller protons. Today, therefore, so there was a transition with the first 60 kilohertz, 1.3 rotors capable of 60 kilohertz magical spinning, in which proton detection was demonstrated with 100% reprotonated deuterated molecule with 100% reprotonated exchangeable sites, and with the advent of Bruker's 0.7 millimeter probe, we entered the new regime with fully protonated molecules, but become accessible to atomic level investigation, conjugating both resolution and sensitivity, because today only 0.5 milligrams are needed for studying a protein sample, with respect to the 20 milligrams, but was the large amounts, but traditionally were needed for solid-state NMR. Now, this is the milestone realization, but so we were recorded in 2015, 2016. So when we saw that indeed the expectation of which the community had built this race toward faster spinning indeed worked for a microcrystalline protein that was GB1, and you can see the improvement in resolution, but also counterintuitively in sensitivity in achieving a two-dimensional fingerprint, the proton-nitrogen correlation, proton-nitrogen fingerprint for this for a fully protonated molecule. So less than 100 Hertz. This is not solution like NMR, but still is a resolution which is sufficient to fingerprint site-specifically a molecule. Now this works because this is a microcrystalline protein, and there was a lot of at the time, a lot of skepticism whether the methodology would work on more complex molecule in different aggregation states with a less homogeneous preparation, like membrane proteins. So in microcrystalline GB1, this is so you can see there is a decrease of the line with mass frequency. So this is the axes are turned, go from right to left. So, and there is a large steady decrease, linear decrease, because the homogeneous broadening is decreases roughly with inversely proportional to the spin to the spin rate, and the homogeneous broadening. So the consequence of the homonuclear dipolar couplings is the dominant contribution to the line width. The inhomogeneous broadening, so the sample disorder, is only a small portion, and in a microcrystalline sample. Therefore, this explains this trend. Now, In the case of membrane protein, this may not be the same. And there was a lot of skepticism at the time whether this same advantage, the same progress would be observed in a membrane protein. And finally, what we and others demonstrated over the last few years is that the same benefit or a comparable benefit is obtainable also for a membrane protein reconstituted in lipid bilayers. So this is the example of the alkyl, so 230 amino acid, so 28 kilodalton, transmembrane protein from Pseudomonas putina, reconstituting lipid bilayers. You can see that 60 kilohertz already protonated deuterated molecules have different line widths, sign that the homogeneous dipolar coupling contributes to the line width, and if you spin faster, you get a decrease in line width, both by faster spinning and by proton dilution, meaning that also in this case, also for membrane proteins in lipid bilayer, the homogeneous broadening is only a small portion of the line width, and there is a significant gain both for protonated and deuterated molecule in spinning faster and faster. This is interesting, which and this opens up the revolution of the fast spinning also to the field of membrane proteins in lipid bilayers. In addition, the fact that line widths are dominated by homogeneous broadening also accounts for the fact that higher magnetic fields have resolving power in the spectrum also for this class of samples. This is another membrane protein, a bacterial magnesium transporter. We'll look at it in a few minutes. Also has a significant reduction in line widths, going from 500 to 1 gigahertz at 100 kilohertz under magical spinning. And the increase of magnetic fields comes also with a significant increase in sensitivity, going from 14 hours to 2 hours to acquire the same fingerprint. Unfortunately, we don't have 1.2 gigahertz data in Beyond yet, but you can imagine. So this is good news for the trend, and this is so since we don't have higher fields, we can just simulate. We don't have higher fields, we don't have higher speeds. But if we know what's the amount of the relative contribution between homogeneous and inhomogeneous broadening in these samples, we can imagine that this sample will have at 1.2 on a 200 kilohertz magic angle probe, will have a multitude of sites fully resolved. So there is no reason to doubt about this kind of improvement. So this class of samples are fully suitable for fast magic angle spinning. What are the advantages of the possibility of studying fully protonated membrane proteins just rapidly? One is the fact that it's possible to optimize the preparation of a sample directly with proton detection on a sample which are only labeled with proton and nitrogen. So this on very small quantities, which makes it very easy therefore, and to screen several conditions, for example, to optimize the kind of the nature of the lipids. You can see here 2 spectra acquired with proteins reconstituted at a given protein to lipid ratio with two different lipids. The quality of the spectrum can be used to probe the most appropriate lipid environment, and therefore to exclude environments where the protein is distorted, aggregated, or inhomogeneous. Then the same kind of spectroscopy can rapidly be used to monitor the protein to lipid ratio, and finally, once all the conditions are set up, this is the spectrum that one gets with the highest magnetic field and highest magic angle spinning rate. So this streamlined sample preparation, sample optimization is much more practical as respect to what was possible with carbon detection in the earlier time of solid-state MR, where much larger sample volumes and therefore much lower optimizations were achievable. So a second advantage of fully protonated molecule is that in transmembrane molecules, well, one can conjugate proton detection with the full analysis of the transmembrane portion of the molecule. This is proteorhodopsin, and Vlad Ladizansky has pioneered the field here, demonstrating that deuteration was interesting in this class of systems because actually one can highlight the water accessible path of the molecule in an otherwise invisible environment, because the molecule, the deuterated part of the molecule, cannot be proton exchanged. And that's the red spectrum. At the under kilohertz, we can look at the molecule in a fully protonated form, and we can therefore not only find again all the resonances that were visible at 60 kilohertz in the deuterated case, but also all the additional resonances belonging to the non-exchangeable transmembrane portion of the molecule previously invisible, and use them to detect contacts at the inside at the core of the protein, before characterizing the fold of the molecule in its transmembrane part. And also here on the right, you can we can highlight contacts between the protein and, for example, the retinal, so the portion, so the cofactor at the core of the molecule, which was invisible in a deuterated case, in a deuterated preparation. And then the third advantage is that we have now two different windows into the protein, a new entry point into the side chain, because not only we have a proton nitrogen fingerprint, but we have also proton carbon fingerprint, so a second window that we can use to monitor the state of the protein, its interaction with ligands, but also to assign resonances. And all this has produced a pipeline, which is finally very similar to what can be applied very similarly now today on a membrane protein in the solid state, similarly to what is done for a protein in solution. So can assign backbone resonances with amide or H-alpha detected triple resonance experiments. A suite of experiments has been developed. You can assign side chains with a carbon-carbon toxic proton detected, and you can finally monitor proton-proton contacts directly instead of the indirect experiments, which were relying on carbon detection, transfer to proton mixing back to carbon, which made the core of solid state NMR for many years. And with all these directly detected proton-proton proximities, we can calculate structure using finally the same software, the same approach developed by the solution NMR community. The example here is the structure that Lauren started calculating when he was in Lyon before becoming a professor, a PI, a group leader in Göttingen. And well, we used here 4D experiments on a deuterated molecule, first to get amide-to-amide contacts, which generates a highly ambiguous series of contacts, which can then disambiguate with the use of more information from fully protonated samples and the algorithm that Sayana has calculated the structure. So after resolution of the ambiguities, there is a very clear pattern for data sheets, and with all the data from a fully protonated sample, the software recognizes hundreds of distance restraints out of which a structure is calculated. So this is the result here on the right, published last year in PNAS. That's fine, that's interesting. And actually the same methodology has been now over the last few years applied on different samples, different classes of samples of increasing proton content from the first microcrystalline proteins, deuterated and reprotonated, where we try the approach. And the first membrane protein, well, Lauren again worked during his PhD at MIT, where he inserted the first proton detected contacts, the first very few proton detected contacts detecting a deuterated molecule. We have we worked with ARPMOT on G, and again there was a mixture of protonated of proton contacts acquiring a deuterated sample mixed up with carbon-based contacts to full proton-based determination in capsid a membrane protein, and the fibril. So we have 3 classes of samples where now the approach has been really proved, benchmarked, and demonstrated. Well, this is fine, but then, so today I would like to add something more to this approach. So what's really special? What's peculiar in a determination of a structure of a membrane protein by solid-state NMR? So membrane protein structures can be determined. Actually, the same protein here was determined here on the right by magic angle spinning in lipid bilayers, but was also determined by solution NMR in mice cells. And actually, an analog molecule was previously crystallized and sold by crystallizing detergents and sold by X-ray. So the structures that one gets are wildly different. And but what's the added value of solid-state NMR? Well, the first point is that solid-state NMR is the structure where is the technique where the only structure where naturally the molecule can be studied in lipid bilayers. So in a reconstitution in an environment which is the closest to the cellular environment. And the second is that similar to solution NMR, NMR carries the capacity to conjugate structure with site-specific dynamics. So the anisotropic interactions present in a sample are modulated by motion, and solid-state NMR has two ways to look at them. So site-specifically, we can measure how the anisotropic interaction is reduced, scaled down by motion, or we can indirectly also simulate what is done in solution, monitor the way in which the motion of this, the modulation of this anisotropic interaction enhances nuclear relaxation and measure nuclear relaxation. Around these two approaches, there are a number of techniques which are developed over the last 20 years. So I would like to advertise here a review that we recently wrote with Marta and Tanguy in our lab for a special edition of the Current Opinion in Structural Biology, organized by Marius Clore, previous speaker. So thanks, Marius, for inviting us. So that's here we try to summarize the different approaches, the different experiments. There is a large palette of experiments based on recoupling of anisotropic interaction, spin relaxation, relaxation dispersion, exchange, cryo-MIS. But I would like to show you three examples where the peculiar capacity of solid-state NMR to investigate dynamics can highlight something new. First one is the sample of aquaporin. It's a collaboration with Vladimir Ladizhansky. So aquaporin is extremely well-known sample. There are many crystal structures. Aquaporin 1, it's a tetramer, and actually Vlad realized that many structures solved by crystallography shared exactly the same transmembrane portion, but a discrepancy in the description of a loop, the loop C, which for which different roles or different hypotheses were formulated for the involvement in water transport. And based on carbon detected NMR data, he recently refined the structure of this loop, and he found evidence based on mechanical shifts and on some carbon-carbon contacts that this loop actually is very well regular and structure. So is this real? Is this an artifact? Do we have other evidence? We teamed up, started analyzing this kind of samples that is produced in yeasts and cannot be deuterated. So it's a class of samples for which fast spinning and full protonation has an undoubtable advantage. With the procedure I sketched above, it was possible very rapidly to get complete assignments of both the carbon and proton HSQC. and to measure dynamics, to measure anisotropic interactions. And then we had to confront with the fact that a very fast magical spinning, actually, the experiments to measure anisotropic interactions, to recouple, for example, proton and nitrogen, needs to be readjusted. The RIDO scheme developed by Paul Schander, Bert Meyer, Matthias for probing dynamics in a highly deuterated environment doesn't work at full protonation. So the RIDO recoupling curves, the modulations cannot be translated into a distance, provided there are additional spins. There is a strong network of proton spins around the proton-nitrogen pair, and we used a CPVC, a variable contact time, a CP with a variable contact time, which is a technique which had already been introduced in the context of carbon detection, slower spinning speed. And we noticed that this technique allows to measure the proton to recouple anisotropic interactions also in the context of full protonation. So with this in mind, we try to go one step farther, and so using concepts also that are already circulating in the community that can be fully realized in this new regime. We designed an experiment that actually exploits the full protonation of a sample to simultaneously transfer polarization from proton to nitrogen and to aliphatic carbons, and to record a proton nitrogen and proton carbon HSQC at the same time with no sensitivity penalty, and therefore to record dipolar couplings of proton nitrogen and proton carbon dipolar couplings. This works well on the model GB1, but what's interesting is works well also on aquaporin. So simultaneously with similar experiments, we could record site specifically backbone amide and backbone CH dipolar couplings, and therefore all the parameters compared with a rigid value. And one can see that all the parameters are very high for helices, are very lower for loops, but for the loop C, the terminal portion of the C-terminal portion of loop C has very high order parameters compatible with the structure, with the NMR structure that blood are determined. And what is more interesting is that the same experiment with the same HSQC, the same carbon HSQC, we can also directly measure order parameters for all the side chains based on the resolved carbon proton HSQC for all the groups, the different groups, the methyl CH2. And for example, we could see that around in the loop C, at the terminal part of the loop C, there is a valin with an extremely rigid side chain, much more rigid than all the other side chains, and that's exactly the anchor used for this loop is the amino acid that anchors the loop C to a hydrophobic core, and that's the mechanism used by aquaporin finally to filter more efficiently water molecules at the entrance of the pore. So this is a new insight that comes associating structure and dynamics in this determination. Second example, Lauren may have already showed that there is a lot of Göttingen contribution there, because these are data that were also co-analyzed with a group of Bertegut in Göttingen. So here we compare, as you can see, in solution, the molecule has shares the same transmembrane structure, transmembrane fold of the lipid bilayer determination, but the loops are completely disordered, either invisible or really disordered, while in solids they form up with this extrusion structure, the extrusion. How important is this extrusion? So there is structure there, but there is also peculiar dynamics. So the transmembrane part, as measured, as confronted with R1 raw, is very rigid, while this extrusion, which is still folded, has very much higher R1 row, nitrogen R1 row, which are indication of microsecond, millisecond motion happening there. And the same residues experience also chemical shift perturbation by interaction with the substrate. And all this was analyzed, again, thanks to partnership with what Laura set up with Verde Groth, so a real deformation of the upper part of the molecule, which enlarges a break in the hydrogen bond pattern of this extension, which is used by the protein to transport the substrate, so small hydrophobic molecules, not across the membrane, but into the membrane. Therefore, to allow to revise the transport mechanism. So this is a molecule finally, which doesn't again transport across the molecule, but uses dynamic to, so this peculiar melt of structure and dynamic to gather substrate molecules, collect substrate molecules, and funnel them into the membrane. So in the end, this is a mechanism to allow polar molecule to bypass the polysaccharide layer in outer membrane, in outer membranes. And then just one point saying that mimicking the membrane environment, so the capacity that solid-state NMR has to look at membranes is, so look at different preparations is an advantage. In this study, the HSQC on the right, fingerprint didn't change between the MPC preparation and preparation including the lipopolysaccharide layer, saying that already the MPC represents a good membrane mimicking environment. But there are other cases, for example, for the OMP-A molecule, I haven't labeled that here. This is a working collaboration with Johannes Thoma and Björn Burman in Gothenburg. The MPC sample reconstituting the MPC shows the purple spectrum, so only a portion of the signals, no signals from any of the loop, while the sample reconstituted in vesicles produced with the same composition of the outer membrane, produced with a special technique developed in Gothenburg, allow recording a spectrum where large portions of the loops are visible. So this technique is particularly versatile and can be adapted to different preparations. The very last examples, the last few slides, where again the capacity of solid-state NMR to describe dynamics are particularly interesting is the CoRe, so bacterial magnesium uptake system. So a very well studied system is a pentamer of overall 200 kilodalton. And there are several high resolution X-ray and cryo-EM structures for the two limiting forms that actually are supposed to describe the transport process. A so-called closed form and an open form. Closed symmetric form in presence of high magnesium, and an open form that was trapped at low magnesium concentration, which is more asymmetric and therefore possibly more dynamical. Based on these classes of structures, the mechanism for magnesium transport is based from a transition between a rigid dry closed state and a flexible hydrated open state. We looked at this molecule in reconstituting lipid bilayers with and without magnesium. We could assign the spectra. It's a large portion of the spectra with 60% of the transmembrane domain and of a alpha beta alpha domain in cytoplasmic portion. And we use these domains to monitor relaxation rates. And what one can see here that removal of magnesium promotes an increase of dynamics compatible with the model open closed states. So with magnesium, peaks are intense, and longer relaxation rates, longer nitrogen R1 relaxation rates. This drastically changes with the removal of magnesium. So this may support the established mechanism, but there is a problem. Actually, looking at the HSQC, the spectra are extremely similar. The changes between the chemical shift changes between the two forms are negligible, both in 2Ds, in 3Ds, with different samples with different labeling schemes. And actually, this closed and open state have also the same SANS profiles reconstituted in solution in micelles. So actually, the two-state model with drastic changes between closed and open is not adapted. So we have played a lot with the sample, and these preparations allow to work on a fully protonated Clore molecule washed in D2O to look at non-exchangeable residues, or look at the deuterated molecule and back exchange to see the exchangeable residues. And actually, we note that the transmembrane pore does not exchange with water, so it stays folded, but is not so dry, because actually the peak intensity in D2O and H2O is changing, witnessing the presence of water molecules, slowly moving water molecule inside the pore. So the transmembrane pore in the closed form is not so dry, and the transmembrane part is not so rigid. Actually, we could measure R1 raw relaxation. There is a gradient of relaxation. Well, in the connecting region, there are only few sparse assignments, but one can see that there are extremely large R1 raw rates witnessing the highly dynamical intermediate connecting regions. So the central region are the most dynamic, and we could also determine for this region in a deuterated sample relaxation dispersion, therefore only for a subset of the residues, exchange the relaxation dispersion that are the signature of the exchange between different conformations occurring also in that structure. So this overall allows to reformulate the concept of the rigid, dry, closed state into a concept of a flexible, hydrated, closed state, exchanging with a flexible, hydrated, open states, therefore changing the paradigm for rigid, dry state, which has a single conformation, to a more dynamic or hydrated state where several conformations are accessible. And therefore, the transition between one of the two states involves only a slight rebalance of the different populations and not a sharp major transition. This is a work that we are carrying out in collaboration with the SANS people and the people in Copenhagen. And I finish here thanking the people I had the privilege to work with in Lyon over the last few years: Lauren, our MPI, Diana, Andrea, Marta, Christophs, Riga, Daniela, Tanguy, Tobias, Jan, now in Warsaw. Again, the people in Göttingen, Vlad Ladjansky, Liz, Kresten, Tone, Nikolai in Copenhagen, Tom, Katrin in Munich, Johannes, Björn, Hartmut, Johann Sebastian Rettel, Vipin, Madhu, and a bunch of people at Bruker that unfortunately are not solving all the problems that the equipment has, but have been wonderful in getting all this revolution with faster revolutions possible over the last few years. And I thank you all for listening. I remind you that the laboratory we have in Lyon is an access provider within French national and European access projects like the IRRMN in France and the INEX Discovery in Europe. So if you are listening and you are interested to try any of the fast mass experiments and many more in Lyon with us, look at the website and submit a proposal, and we will be happy to work with you.
Thank you, Guido, for this nice update and progress report about the power of ultrafast proton-detected MAS NMR, particularly on these exciting membrane systems. So we have a several questions already lined up in the Q&A. The first question is about rotor packing from Christoph Grohe. Thank you for the great talk. For rotor filling of membrane samples, are you using an ultracentrifuge?
Yeah, so in the end, that's the least problematic, the smallest of the problem at the moment. This is quite standardized. So we use an ultracentrifuge. We have we and the community. I mean, there are even commercial a number of filling tools, and so that does not produce any major problems recently.
Let me ask a sort of a related, a different question. So have you seen any sign of membrane proteins behaving somewhat differently under 100 kilohertz MAS versus more benign or slower MAS, especially for these extramembrane domains or peripheral regions that might be, is it possible that they might get distorted, structurally distorted by spinning so fast?
Yeah, so it's a pertinent question. We haven't observed any neither degradation of the sample, nor decomposition of the membrane, nor sign of. So we have in particular for some of them studied so fingerprint spectra at different spinning speeds, because of course, so you cannot compare a small rotor spinning fast. Spinning so a 3.2 spinning at 20 kilohertz still has a large forces. So also in that case, maybe I mean, there might be distortion. So we have followed up the phenomenon for all the samples I showed you, many more. We haven't seen any sign of that.
Yeah, especially the hydration aspect. If you're spinning that fast, I'm just wondering if the water will get all pushed to the middle of the rotor and the membranes will be drier. And since you are looking at some of these channels or hydration aspect, that might be something to watch out for.
Yeah, so provided the sample is watertight, provided the rotor is watertight, we haven't seen any degradation.
Zhi-Yu-san asks, have you tried deuterated lipids and will it make a big difference to the quality of the spectrum?
Yeah, so this, there are samples where there are cases in which we try deuterated lipids and the effect is important depending on the sample. Yeah, this is one of the components, one of the elements to screen indeed.
Okay. So Marty Ninot Pedrosa asks, says many thanks for this great talk. I saw that you work with non-deuterated lipids. Do you think they're useful to improve signal? So I guess this is the similar question to the last one. Okay, so let me see. Let me move on to another question from Christoph Grohe regarding proton-proton distance restraints from your CP-based approach. What are the advantages and disadvantages compared to finite pulse RFDR for measuring proton-proton distances? How do you calculate the distances?
Well, there is a universe there, but is so you cannot expect that this is a quantitative measurement of extensive set of distances with high precision. So I mean there are several subsets of the questions there. So the first portion was about RFDR versus bus spin diffusion, and so it's the bus spin diffusion. So it's a chemical shift selective band selective recoupling, band selective spin diffusions, which therefore allows to get with high intensity and restraints between amides, for example, in fully protonated samples, or between methyl groups with a fully protonated sample, or between H-alpha protons in a sample where otherwise magnetization would flow away in several directions, and also to win dipolar truncation. So the advantage is considerable. The RFDR is what we use typically in a deuterated molecule, where the need and for bus spin diffusion is not is less stringent. As for the conversion into distances, as I said, but that's a pot that is better not to open. Cyana has an algorithm to do this translation. But what is crucial is to be able to establish a fold, an initial fold. And for that, we use the fact that we use chemical shift, we use talos in the beta barrel, for example, to get the strands. And then if in a strand or between two strands, we do see a contact between the corresponding amides, we impose a nitrogen bond. So if there is evidence for a standard conformation and there is a contact in the RFDR or in the bus, then we impose the hydrogen bond. This helps in fixing the initial fold, and then Cyana has a protocol to determine the convert volumes into distances.
Mm-hmm. So related to this, a quick question is, do you have a sense of the maximum distance that you could get from proton-proton measurements? Or has it been calibrated at least?
Yeah, so that's, there is a calibration. So the calibration is done intrinsically in Cyana. The problem so there is always this question. So what is the maximum distance? So I think that there is not a problem of distance here, but there is a problem of that by increasing the distance, you increase the ambiguity of the constraints. So finally, the constraint is not considered and not used by Cyana or not interpreted. So in order to do this calibration, one should really prepare a sample with the sparse pairs of protons and then measure the distance.
Yeah. Okay. So here's a biological question from Rams. Did you also measure proton-proton NOEs and well, okay. So sorry. Let me switch to the other Rams question. Great talk, Guido. What factors contribute to the structural differences for the extracellular loops? Is it the beta barrel protein that you showed from micelles and lipid bilayers? Is it the curvature of micelles or the lipid loop interaction? Do you observe any differences for the transmembrane regions?
So transmembrane region is pretty similar in the two cases. Okay. I imagine, so high ramps. So I show here the two molecules. So I imagine is talking about this figure. So the transmembrane region is pretty much conserved. So the differences are in the loops. And so if we think of this molecule, so in this specific case, the problem may be related on the fact that the detergents interfere with the extracellular fold. So this is a molecule that is shuttling hydrophobic molecules across the outer membrane protein, so across the lipopolysaccharide layer. So in the polarity of this extension is opposed as respect to the transmembrane portion. So there is the polar is outside, the non-polar is inside. So detergents may interact with this portion and induce unfold it. Apart from the fact that this molecule, so this portion of the molecule is dynamic, we conserve the structure in the solid preparation, and this structure may be exacerbated in solution preparation and may render most of this portion invisible.
Mm-hmm. Okay. So there's still more questions for Guido, but since it's 11:30, I would like to move to the informal part of the today's program.